Okay, hello everybody. It's a pleasure for me to present this talk, and it's a challenge in a few minutes to present a large overview of MSC-based therapies, and also I would like to share with you two achievements from my lab. So, first of all, to understand the role and the function of MSC, it's necessary to take a step back. Indeed, any function in any organ is a result of a strong interaction between two cell compartments. The first one is a parenchyma, which contains the specialized cells, such as cardiomyocytes for heart, neurons for brain, and stroma. Stroma is a ground for the specific compartment of parenchyma. And with such picture, you see that MSC can be found in both compartments. But on one end, in the parenchyma, they can behave as progenitors for specialized cells, for adipocytes, for cartilage, for osteoblasts, for tenocytes. And this behavior is only in mesenchymal tissues. On the other hand, you can find mesenchymal stem cells in all tissues, in all stroma, such as a network which is responsible to generate, to structure, and to stabilize the stroma. And so why and how MSC are able to do this job? Because in fact, they have many functions. Of course, they are multipotent. The name is stem or stroma cells, but also they display a great paracrine activity. And through these different activities, they are able to manage a lot of things into the stroma and into all tissues. The first point is they are able to manage the inflammation and the immunity with a capability to interact with all immune cells. They are also able to manage vascularization through the secretion of angiogenic factors, but also through their capability to behave as pericyte and to stabilize vasculature. They play also a great role in the structure because, in fact, they give rise to fibroblasts. They are responsible to secrete extracellular matrix, and they are involved in their remodeling. And finally, also, they have a great capability to handle the metabolism through their capability to differentiate into adipocytes. So, through all of these functions, they behave as a true conductor for all organs. And so with such picture in the mind, if you think about MSC-based therapies, it's necessary to consider two quite different paradigms. The first one, of course, is to use MSC as progenitors for specialized cells. It's a first option. The second option is to use them to indirectly recover or to indirectly correct some tissue dysfunction or some function impairment. And so you can use MSC for their role as supportive function, and a role as conductor for any tissue. And in fact, if you try to make a picture of the clinical trials in the world, you see that most of clinical trials in the world in the field of MSC are clinical trials devoted to the function of the supportive function. And you find different clinical trials involved in quite a large number of applications and diseases, and you see here the use of MSC as mesenchymal precursors, and you see that the number of clinical trials is relatively limited. And if you go through the literature, it's clear that MSC are very effective, for instance, in GVHD. In Crohn's disease, you have a market authorization in this field. You have promising results in intervertebral disc and also in critical limb ischemia. So all context corresponding to the context of supportive function. So it's a picture of MSC. Now, how we can move forward? In fact, the results obtained with MSC are relatively variable. And so it's necessary to try to have robust effect. And so to do that, we think about the fact to generate a therapeutic 3D mesenchyma or a therapeutic 3D stroma in order to correct the dysfunction in the tissue. And to make the story short, we made a proof of concept, in fact, in an emblematic disease, which is an age-related chronic inflammatory disease associated with dysbiosis, which is periodontitis. And in fact, we set up a clinical trial in aged dog that display, aged dogs are very relevant model because they display exactly the same symptoms and the same feature than the disease in human. And using aged dog and using a strong design, which is a randomized double-blind controlled trial, comparable to a human trial, we were able to demonstrate that if you bring ASC, the network with hydrogel, we are able to improve inflammation with a decreased bleeding. You improve the growth of the tissue, and even you correct the dysbiosis. The correction of dysbiosis perhaps is not permanent. We are investigating this point now. So you see that if you are able to bring ASC in the correct form, you can have a strong result. The second achievement I would like to share with you is a different achievement. It concerns the use of MSC as progenitor cells. And to do what? To do adipocytes. And you can ask, adipocyte, what is the interest? In fact, in the field of metabolic disease, there is a very interesting adipocyte. The name is beige or brown adipocytes, because such cells is able to burn energy, and in fact, it's a way to limit the energy storage and to limit obesity and metabolic disease. And again, to make a story short, here, in fact, we set up a bioprocess suitable to produce organoids with our very relevant model to screen all drugs you want, but using the same process and by scaling up, we are able to generate a 5 mm object, vascularized, which is suitable to be transplanted in order to correct any metabolic disease. So now, presently, we are investigating the result or the consequence of the transplantation of such object in obese or diabetic models. And so, just to keep the time, I think I was perhaps too short, but here you have the conclusion. I think MSC, this is not the end. We have great hope in MSC. Okay, it's different from CAR-T, but you have very challenging field, and okay, we believe in this field. But it's necessary to have a new understanding of MSC. It's necessary to have a new look on MSC and the fact to use MSC. How we think about MSC, I hope that I convince you that 3D models are very relevant, and it's easy, relatively easy to get and easy to manipulate. And of course, I don't speak about exosome and engineering MSC, which is another topic, and the time was too short to speak about this. But this domain is also very active. So, thank you.
Thank you very much. Thank you.